So during today's lecture, we will now focus on the metabolomics workflow. And here, as a background information, I have put into the Moodle site this article called NOTAME, Workflow for Non-Targeted LC-MS Metabolic Profiling. And this article is the one paper that I would recommend you to read in terms of how to set up a metabolomics pipeline. And this is an article that is based on our own research group practicalities, all the information starting from very beginning of the metabolomics workflow all the way towards the result interpretation then. So this is now included in the Moodle, and this is something that I expect you to read in addition to these slides. And this is also something that will be later on during the data analysis part used during this course. So yesterday we already took a look on yesterday on Monday we already went through a little bit on the experimental design and sample collection, and here I would like to just remind or repeat once again that any biological sample can be used for metabolomics analysis, and the study setup and the study question of course then depends on what is your focus. So you can use whatever, for example, human biofluids, or it can be an animal-based sample or a food sample or whatever biological sample. And the experimental setup is then based on what your study question is. And on that part we don't focus any further during today. But let's go then through the sample preparation, how to actually conduct the metabolomics analysis in the wet lab basis. So the aim in sample preparation in metabolomics analysis is that first of all we want to get rid of other chemicals than metabolites from the sample. You might recall that on Monday I mentioned that metabolomics is basically or you can address with metabolomics all the chemicals after you precipitate proteins, polysaccharides, and any genetic material from your samples. So first thing is that you need to get rid of those that are harmful or that might hinder the analysis or the detection of metabolites. And then, on the other hand, you want to get rid of others than metabolites, but also you want to keep in your sample as large number of metabolites as possible. So in terms of a number of different chemical structures, so that should be always maximized in metabolomics analytics. And also those various different chemical structures, they can be present in the sample in very different levels. So the concentration levels can be several orders of magnitude difference, and that is an inherent point also to take into account in sample preparation for metabolomics analysis of in general the whole analysis. And also when going for sample preparation, you need to be aware that solubility of the compounds may vary. So how can you keep there as much polar and non-polar compounds as possible when you know that polar metabolites, those only are soluble in water, and non-polar, the more fat-like components, they require very strong solvents to be dissolved. So where is the balance here, and how can you maximize the coverage? And still one additional focus point is to quench the samples, meaning to stop any metabolism that is occurring in the samples. And this is especially relevant if you are taking, for example, a time series from some experiment where you, for example, induce some metabolic reaction, let's say in a cell culture analysis, then before you go for the extraction itself or any analysis, and if you want to compare those different time points, you need to be sure that you stop any metabolism that might be occurring. This is actually easily achieved if you use, for example, some solvent that precipitates proteins. So that's already taking quite a big part of that issue, so that no enzymatic reactions will be performed anymore when proteins are precipitated. And also, in the same time, when you want to get as wide repertoire of compounds, you also wish to keep those compounds in as intact form as possible. So the more metabolites or compounds you get from your sample in non-degraded form, the more likelihood you have that you can actually identify those compounds in a correct manner. So for these reasons, sample preparation and selection of the correct means for sample preparation in metabolomics analysis is a relatively essential step. And further highlight the fact that in metabolomics analytics, we are looking at very extreme chemical diversity. So for this reason also the solvent selection is absolutely important, because that will determine what is the pool of metabolites that you can actually get in your sample. And here we need to distinguish metabolomics analytics from other omics, especially genomics and transcriptomics, because if you think about, for example, gene expression profiling, there you are basically catching the RNA molecules. So RNA, that is a very sort of restricted structure, chemically speaking. Only the order of the bases is altered, but you can easily extract all the RNA and focus your analysis there on the whole composition of what have been expressed from genes. But that is not possible for metabolites because of this variability in the chemical structures. And here just to distinguish a little bit what kind of components belong to which side. And many times when speaking about very, let's say, a general metabolic profiling, or first assay that you typically conduct in metabolomics might be something that you focus on what we call semipolar compounds. So that would be something in the middle of that red arrow. So phenolic compounds, alcohols, also some amino acids, and this sort of constituents. So we typically are in relatively on the what we call semipolar range, not in the extreme polar or extreme apolar. Those we need to have specific analytics to focus on. So to conclude, there is no unique extraction protocol that would cover all intracellular metabolites, and combination of several extractions is needed in order to increase the coverage. If you want to gain as much knowledge on your sample, then you need to have several different sample preparation methods. Here are a couple of very simple ones. And the first protocol here, so this is the simplest one that you can have, and actually this is what you will be also doing during the course. So you have your sample. It can be readily in liquid format, or you need to homogenize your sample if it is in solid form. For example, of food samples, you need to homogenize it in solvent. And then you add the chosen solvent. And after adding the solvent, you will see forming of a precipitate when, for example, proteins will be precipitated. Actually, this will not be the case when you will be analyzing your own urine samples, because naturally in urine we don't have proteins. But nevertheless, the solvent is added there to get rid of if there are any impurities or so from the environment, and also that is needed for the sample to be injected to the instrument. After you have added the solvent, you see the precipitate, then you centrifuge your sample, filter it, and then it is ready for analysis. So this is the simplest way to do metabolomics sample preparation. And as an example, if your sample is already in a soluble in a liquid form, then, for example, plasma samples we typically prepare so that we add acetonitrile directly to plasma, so that we have 4 times more acetonitrile than plasma. And in case of urine, that ratio can be something like, for example, 1:2. On the other hand, then, if we go for extraction of metabolites from tissues, very commonly used procedure is to add 50 to 80% of methanol to the samples. And why not 100% is that we also want to have the possibility for those a little bit more towards water-soluble compounds to be seen. So if we would add 100% methanol, we would already then put the extraction towards relatively non-polar compounds, but we also want to push it a little bit more towards the semipolar region, and that's why then it's good to have a little bit of water also in your extraction solvent when you are dealing with tissue samples or food samples, for example. In addition to these very straightforward, simple extraction methods, there are also various more complicated procedures that are required due to the choice of the analytical approach or due to the choice of compounds that we want to address. For example, in gas chromatography, it's very typical to derivatize the analytes. So not all compounds are readily evaporated. They cannot be analyzed in gas chromatography unless they are evaporated. So in order to increase the number of compounds able to detect there, they can be derivatized with different procedures in gas chromatography. Or then it might be that you have something in your sample that you want to get rid of before going for the actual extraction. In that case, you can, for example, perform solid phase extraction so that you add your sample to this kind of SPE pre-column, and then you collect your sample after the harmful substance is bound to the SPE column. Or it can also be vice versa, so that you are interested in those compounds that are bound to the column, and in that case you only take those components that you can then take out from this pre-extraction column. And what is currently seen in increasing amount in literature are these multi-phase separation methods, where both the water-based and solvent-based solvents are added, so that you can have first, for example, here is the one such a scheme that you remove the precipitate, then you add polar solvent and you get 2 different phases, and these 2 different phases can go then to different chromatographic separations or different mass spectrometry analysis. So you can distinguish those. And typically, if you do this kind of multi-phase separation, then you try evaporate the solvent and then redissolve it to a suitable solvent that can be then injected to the analysis. But this evaporation and redissolving, it's not always necessary. So the samples can be already analyzed as such without this additional step. But this kind of procedures are increasingly reported in the literature when you really want to maximize different pools of compounds that have different chemical properties, that they either go to the water phase or then to the solvent phase in the extraction procedure. And once you then have your sample processed with these, whatever your selection for the procedure is, then it has to be analyzed with one of these. So for example, now we will be mostly focusing on the liquid chromatography and mass spectrometry analysis. Next week you will hear then what these procedures are if you want to focus on NMR-based analytics. So Maria will share that information. But now let's see how LC-MS can be used for metabolomics purposes. So first of all, separation of analytes with chromatography. So I'm sure that all of you have encountered the definitions and the principles of chromatographic separation already earlier, but just to remind, in chromatographic analysis, the compounds are separated based on interactions of analytes in your sample with the mobile and stationary phase in your analysis. So stationary phase is the matrix that is embedded in the chromatographic column, either gas chromatography or liquid chromatography, and the mobile phase is then the solvent that is pushed through the column with high pressure. And your sample is then in such a form, in such a solvent that is compatible to be analyzed with in this taken mobile phase. So whatever is included in your sample has to be separated so that you will maximize the likelihood that you can then detect the different components. If they are all in one big clumps without any separation, then you will for sure lose information from many of the components. But chromatographic separation is conducted so that you will maximize the ability of the compounds to be seen in the analysis. And the mobile phase is eluding from the chromatographic column at different time points, and then also the analytes are eluding from the column based on their physiochemical properties. So they have different affinity for the chosen column that is used, and based on this, then they come out in different time points, and we can see different waves coming, or different signals coming from the analysis. And the way how these the elution of the compounds is then detected, there are various different detectors that can be utilized, but those that are most commonly used are, for example, the UV detector. Ultraviolet light absorbance can be used in order to visualize the metabolites coming out, or especially when the data is going towards metabolomics analysis, it is typically combined with mass spectrometry analysis, so that we are not only seeing the elution time of the different compounds, but we also push those through mass spectrometry analysis and detection thereafter. And still just one screenshot. So this is a basic chromatogram, how it looks like. So as you can see in the x-axis there is the retention time, so the different time points when different analytes have eluded from the column, and the different peaks in this chromatogram represent different chemical components. In this case, the detection was conducted in mass spectrometry analysis, as you can see. So it's TOF-MS. Electrospray ionization in positive mode in this case, how it was then analyzed. But the chromatographic separation is very evident here. It's much more easier to look at the components when they are first separated from each other. And here a little bit more look on the chromatograms. So similarly as the sample preparation is crucial in terms of what components you can analyze, the same goes also for the chromatographic separation, both in liquid chromatography and gas chromatography. And here are, for example, 3 different examples from liquid chromatography. So different columns these columns have a very different stationary phase, so they bind very different compounds, and you can have a different portion of metabolites analyzed from your sample. So for example here, what is shown is see the pointer, okay, but the one that is up is from a chromatographic method called hydrophilic interaction chromatography. So this is an analysis that is suitable for water-soluble compounds. So for example, samples in urine can be easily analyzed with this method, whereas then the two lower chromatograms are from reverse phase column, and reverse phase columns are typically used for a little bit more semipolar or even non-polar purposes. So for example, the one that is in middle, reverse phase C18 column, that's the one that we typically use for general metabolomics analysis. So it's a very good compromise between semipolar and non-polar compounds that we can detect there. And then the reverse phase C8 type of column, for example, the lowest one here, that is suitable when we are especially interested on the lipid portion of in our sample. So for example, here in the middle you can see that there is a big clumps at the end of the chromatogram. So that is representing lipids in the sample. And if you put those to C8 column, then you can better separate. So this part is now the same that is at the end of this C18 column. So C8 is suitable for lipid analysis and typically the choice in lipidomics setups. And also in chromatography, the settings that you can change is obviously then the runtime. Traditionally, the liquid chromatography columns have necessitated something like 30 minutes run, but nowadays what is often used are these ultra high pressure liquid chromatograms, which tolerate much higher pressures than the traditional ones, and the benefit there is that you can get as good separation as the traditional columns, but you can squeeze the analysis to much shorter run times. Even something like 5 minutes run is possible. What we typically apply in our everyday analytics is around 15 minutes. So that's a very typical runtime in metabolomics analysis. So to summarize, why we need chromatography in metabolomics, or why do we want to apply it? So clearly the aim is to separate and retain the complex mixture of metabolites in a sample. So we want to get them separated as much as possible so that we can catch or then later on detect as many of those as possible. And also what we need to have during or take into account is that the chromatographic method that we design needs to be reproducible, because typically in metabolomics analysis we are analyzing hundreds of samples in one go, so our sample sequences can be very long. Then we need to have the method reliable so that every time when a new sample is injected, we can trust that similar kind of response will come out from the column, so that it is reproducible. And also in terms of maintaining the retention times correct, and also the signal intensity level should be staying same. And also additional point of consideration is when choosing the mobile phase for chromatography. And here we need to remember that after the chromatographic separation, the sample goes to mass spectrometry analysis, and the analytes need to be ionized in mass spectrometry. So the mobile phase should be such that it is promoting high ionization efficiency for as many metabolites as possible. And here the generally applied solvents such as acetonitrile or methanol, they are perfectly good for this purpose. But in some cases, if you are focusing on some particular compound group that is not ionizing efficiently, then it might be that already during the chromatographic phase you could have some adjustment on the sample preparation and the analytical conditions so that you maximize the likelihood that these components can be ionized. And of course here then there are many analytical challenges as in any analytical chemistry method, and when it comes to chromatography, those that we typically encounter are first of all the instrumental drift, so that no matter how you have adjusted your conditions, still you can see that the signal is not reproducible, so that there is a drift that is caused by, for example, the temperature in the room is changing, the mobile phase composition is not remaining the same, the pressure is giving a lot of demand for the instrument, and also then there might be something inherent in your sample that can be causing drifts, and also then gradually the more samples you run, the more dirty your column gets. So there are a lot of sources for encountering various drifts during your analytical run. And also then typically no matter how carefully we try to design the gradient for the chromatographic run, it might be that your sample still contains a lot of what we call isomeric species, so they can be either regio stereoisomerism or stereoisomerism, so that the compounds are having the very same molecular mass and they cannot be completely separated in the analysis. So in these cases you often, when you collect the data, you are actually collecting the information from more than one compound, even when your aim in chromatography is to separate all the analytes from each other. But if you have a lot of isomeric species in your samples, then it's challenging to get those separated properly. Please go ahead.
Is it also possible as part of this during your separation you can have costs it's based on previous analysis done if the equipment is not properly?
Very often, yeah, that is absolutely one of those challenges. Yeah, so it is not only that even if you take a fresh column for your instrument, it might be that somewhere else in your instrument there can be contamination that is resulting from some previous runs. So there are some very nasty polymers, for example, that can cause harm in the analysis several weeks forward. And that is the reason why we typically always, when we start analyzing a new sample type, we first take a couple of test extractions and test runs, put those into analyzer and see how it looks like. Is there a lot of contaminants and something that you are afraid of contaminating the instrument before you can then decide that is your sample preparation and analytical conditions good to go. So yeah, it's absolutely true that there are a lot of steps where you need to pay attention, and I'm sure guys during the lab course will then tell you a little bit more about these aspects as well. And then once we are sure that our chromatographic method is good enough to go, then what about then the mass spectrometry side? What do we need to take into account in this aspect? So I guess also you have had earlier courses on the basis of mass spectrometry, and we are not going any deep detail here. So here are some references, some very straightforward, simple YouTube videos also for the chromatography part, included some links like that. So you can check these if you are in doubt that you missed the main principle here. So in mass spectrometry, we are looking at, we are measuring small molecules. So my colleague always typically introduces mass spectrometry. That mass spectrometry is a huge scale that is used to weight very small molecules. And that is basically true. So it is all about weighing the different molecules. And the molecules can be either directly injected into the mass spectrometer, which is called direct infusion, or through a coupled chromatographic system. And in metabolomics cases, it is almost always coupled with chromatographic system. And I'm sure you can already guess the reason why. That's because we want to get as many components as possible, so we need to get them separated as efficiently as possible so that we maximize the ability of the compounds to be seen. And these direct infusion approaches are typically used in a case when you have a targeted approach. So perhaps you remember from Monday's lecture that we were discussing about targeted approaches, so that you have some target analyte that you want to catch, and you know the mass of that compound, and you are sure you can analyze it without any chromatographic separation, then this direct infusion can be used also. But in metabolomics, in majority of the cases, it is coupled with the chromatographic system. And in order to a chemical compound to be visible or detectable in the mass spectrometry, those compounds need to be in ionized form. So a very crucial step in metabolomics analysis is the selection of different ionization methods. So you need to select from various different ways to transform your compound in a charge form. So what is very often used in metabolomics is the electrospray ionization that is explained in this green part of the text. But what it does, it has an, as the name says, electrospray on the component, which then makes the compound to take us either positive or negative charge, and that makes then the compound visible for the mass spectrometer. And how the compounds then are detected by the mass spectrometer? The ions are detected and reported with a value that is called the mass to charge ratio, or mass to charge value, m/z. So this is what you always see in metabolomics data. And what it means is that you have the mass, and you need to remember it's the ion. So it's always either +1 or -1 from your actual molecule, because it's ionized, so there is one proton added or one proton taken out. And then over Z is the charge, so that whether the component is multiple charged or single charged. Majority of the cases the charge is only 1, and in that case it is m/z, so ion mass divided by 1. So typically, majority of the cases, for example, here you see compound with 665.15 mass over charge ratio, which means that this is a compound with molecular weight of 666. If this is negative mode data, actually that is not visible here in this field, but if this is in positive mode, then it means that this compound would be 664, the molecular weight. And in majority of the cases there is only one charge, but exceptions also exist. In some cases, some particular compound classes, especially some for example, very large polymeric compounds, they can be such that when they go through the ionization, several different positions of the molecule are charged. And in that case, the compound is termed to be multiple charged. And then in those cases, this Z, how the mass is divided, is not 1, but it can be 2 or 3 even. And in these cases, then naturally the signal that you would see is only half of the value for that particular compound. But then, how can you know when it is double charge or single charge? Then this isotopic peak ratio. So as you can see, this peak has something in the apex, something in the middle, and something in the low in the majority of the cases. And these additional values are the isotopic peak values for this particular compound. And in normal single charge molecule, that difference is typically 1, but if it is multiple charge, then it can be 0.5 because it is divided by 2. So that's the way how to detect multiple charge. But that's quite rare cases, and we are not going to in detail of that complicated examples during this. But it's just good for you to know, so that it is, even when it is mass over charge, the charge typically is 1, but exceptions exist. And other issues that you should know about when looking at mass spectrometry data. So this is a spectrum of that particular compound or a taken time point from your analysis. And terminology exists, for example, base peak. Base peak is typically the peak that is highest peak in the spectrum. So for example, in this case, this spectrum base peak is this 665. And whereas then molecular ion is the peak that is representing the intact molecule, so that you have the compound as a complete form, only modification is the ionization, so that it has either proton added or removed. And, for example, in this case, it might be that this 665 is the molecular ion, or it can be any other of these components, but that's something that you will only resolve then during the further data analysis when you are trying to identify the compound. And isotopic peaks I already mentioned, so those are those peaks that come in exactly the same mass, but only 1 mass unit difference typically. If it is 0.5, then it means that it's a double charge molecule. Also, in the case of metabolomics analysis, we are almost never analyzing only the signals from the pure compounds. So that would be the optimum situation, of course, that everything you see in your data would be actually resulting from pure actual individual chemical compounds. But that's unfortunately not the case. But the components, they might have various different adducts. So for example, formation of a sodium salt is very typical, so that actually it is taking not only the proton, but it can be, for example, ionized with sodium, so that you have sodium ion added to the mass. Or it can be in negative form, it's typically chlorine ion or something. So you can have various different adducts. And also in some cases you can have dimers, so that some compounds, they like to bond with actually various different ways of chemical bonds can be resulting to dimers during the ionization, so that it's actually your peak is not representing only 1 molecule, but it can be actually several units. And this you can then resolve later on when the compound is identified through the MS-MS analysis. Or it can be double charge, as I already mentioned, but also the component can have a phenomenon called neutral losses or in-source fragmentation. And this kind of cases, typically then the molecule is not kept in intact form, but it is very sensitive, and for that reason it is then degraded somehow during the analysis. And this is of course very unfortunate situation, because that makes a lot of harm for the data analysis when you see signals there and actually none of the signals are representing the whole compound. But you need to be able to put the puzzle together and actually then think about what is then the actual mother ion, or the original compound there. I'm looking at the time now. Do you want to have a break, or should we go straight ahead? Break. Okay, we can take a break. So this is a good location to break. So let's continue after, yeah, 1:15. Sample. And the compounds in mass spectrometry in the analysis, those can be measured either in the so-called full scan mode. So what I just described to you is the full scan mode, so that it is the compounds are taken into analysis as a complete whole entities. But as you can imagine, in the world there are multiple different components that share the molecular weight. So even if we collect the exact molecular weight from a compound, that's not enough yet to tell actually what is the initial structure of that compound. And for the reason to be able to identify and elucidate the actual chemical composition, the molecular structure of any compounds, we need to utilize also an approach called tandem MS. So this means that basically that the compounds are, in addition to being ionized and analyzed as a whole, they are also ionized and analyzed as parts of the compound. So in principle, the compound is put under a little bit higher ionization, or called collision energy, so that the conditions in the instrument cause the compound to break down to different parts of the molecule. And these bits and pieces of the molecules are then used in the structural elucidation, where you try to put those together and compare the fragmentation pattern with databases and try to get identity for the compounds that you have seen in your metabolomics analysis. And here already now I want to open up a little bit this terminology in the different kind of tandem MS approaches that are used in metabolomics, since nowadays this MS analysis, it is included as part of the metabolomics analysis, I would say always. So if you see abbreviation DDA in terms of MS fragmentation, that refers to data dependent acquisition, meaning that the MS data is gathered or acquired in a data dependent manner, meaning that the precursor ion, it's always known, so that in the scanning phase a certain mass to charge ratio ion, certain compound is chosen for the MS fragmentation analysis, so it is dependent on the actual data that the instrument is recording. And typically in this kind of approaches, something around 1 between 4 different ions can be collected for the fragmentation during a single spectrum or during single precursor ion run. So here you know what is the mother ion, and you can link that directly to the daughter ions then. So what is the source and what are the fragments? Whereas then in approach called DIA, which is data independent acquisition, here the approach is such as the name says, it's not depending on what you have there in your scan, but basically everything, all the ions are pushed through high collision energy, and it creates loads of various fragments in each of the scans. And there the only definition is that you have a defined isolation window, so that certain mass, certain size of molecules are entering the DIA on one go. And the downside of this, or the plus side of this, is naturally that you get a huge range of compounds fragmented already during the during one single MS-MS analysis, so you get a huge amount of fragmentation information. But on the other hand, since the analysis is not data dependent, depending on your source data, you don't have any direct link that which of the ions that are in this particular scan are then the sources for particular fragments. So that means that during the data analytical phase, there are certain challenges or additional challenges that you need to take into account than if you would have done the data dependent MS acquisition. So these are the two most commonly used MS approaches in metabolomics analysis. And if we then take a look on why this is actually done, what is the usefulness of this fragmentation pattern or how it is utilized, a very traditional way of using is to look it in a manual manner. Here is an example of a spermidine derivative compound. The structure you can see here on the right hand side. And this particular compound group is such that there is this spermidine backbone and it is decorated with phenolic acid moieties. So this was an exercise where really on a manual exploration you had to look at what kind of fragment, when you know the size of these different moieties, you know the exact molecular weight of all these phenolic acid moieties, then by looking at what is the size of the fragment, how much it is losing, you can then determine that which kind of pieces are then taken away from the compound. And for example, in this case, the molecular ion, the intact molecule is seen as a low level or relatively small signal ion, because this is from an MS-MS analysis, so the molecular ion is not longer the dominating ion, but the fragments then are. So for example, even the base peak is one of the fragments in this spectrum. So in some cases you need to spend quite some time on looking at the spectrum and really calculating that what kind of pieces there are and how much the molecule is losing from different sides, and then compare that against different databases and try to elucidate the structure. And especially in the case of plant-based compounds, as it is here, many of those have also ultraviolet absorbance. So if you have a device that has, in addition to the mass spectrometry, also the UV detector, it's a great benefit, because the UV detector can directly tell you about what metabolite class you are looking at. And then on the mass spectrometry side, you can have the detailed fragmentation pattern for each of the components.
So just to recapitulate, so at what point do we choose what protocol, as in DIA or DDA? At what point decide to say, okay, I'm going to use this approach, or I'm not going to use this approach? What drives it? What influences the choice?
Yeah, that's a very good question. And so both of those have benefits. So when you have the data dependent, so DDA approach, then you can always be sure that you know what the precursor ion is. So then, for example, in the case of here, you are sure that since this is data dependent and you know that the precursor ion is the 630, so that is already inherently written in the analysis, then you know that all the signals that you see here, they are coming from this particular precursor ion. And then it's much more easier for you to then put those together as one entity. But the downside here is that you are restricted in terms of how many this kind of signals can you collect in one cycle. It is restricted to something between 1 to 4 typically. So you only get very little MS-MS fragmentation information. On the other hand, if you use the data independent approach, you will be able to catch the fragmentation information of all the signals So if we would say that this is a full scan analysis, all these signals would go to the MSMS fragmentation, and you produce a huge amount of fragmentation information already on one go. So there you gain much more information, but then on the other hand, you also produce a challenge for yourself on the data analytical side, that how can I match these? So it's a little bit balancing that. What I would like to do, unfortunately our method yet is not, I would run both, so that I would gather the data that I know for sure, but then if in addition I would see something that is interesting there, then I could also have the possibility that I could go and look for additional fragmentation information. So it is a matter of sort of which is most important balancing in the. But also had to say that, as you asked, that when you decide that, then it's very typical that particular metabolomics laboratories, they have their established methods and established routines of doing analysis. So there are surely laboratories in the world who do completely different way, but these are the most commonly used approaches. So, for example, in this case, we would have the data-dependent fragmentation, so that we know for sure that this is the precursor and it is spliced in this manner. And here, as I mentioned, we use the manual interpretation and try to put these bits and pieces together. So it's very challenging, but it's also very rewarding when the picture starts to open and you get good matches with your. Yep, go ahead.
So you said that you know the precursor already. So does it mean like we have referred to some literature done on this this experiment and we know the precursor, or should we not?
You mean the number of that it is? Actually, that is when running the MS analysis and when running the DDA MSMS run. Then the instrument is telling it. I just noticed that I don't have that information. It should be included in this figure. But the instrument tells, the data file then has that information, that it is telling you that here I used iron 630 as a precursor and here is what it gets. So typically in this analysis, the instrument itself is selecting the 4 most abundant ions in each run. So it is data dependent in that way. So it looks at the data and then does the runs. So that's the way to do. But also then I have to now mention that naturally in mass spectrometry analysis, if you know your candidate compound, so if you know that you are interested in looking at if your sample has ferulic acid, so you know the molecular weight 179 something. Ferulic acid, you search for that and you ask your instrument to produce a fragment ion for that particular, so that please go ahead and search this compound and then splice it into MSMS. So you can have that completely as a targeted manner. But when we are running a metabolomics analysis, we don't have that information in beforehand. So in many cases we are running it completely without hypothesis. So it's data dependent, so we just let the instrument show the data and then choose whatever is most abundant there typically in majority of the cases. Yeah. But there are a lot of different ways. And thanks for the good questions. So and then in addition of doing this manual calculation of the fragment ions in your spectrum, what we often and most typically do is we compare the spectrum against some databases. One of those is called METLIN. METLIN used to be a really nice database because it was free. Nowadays they have started to go a little bit towards more commercial way of functioning, and nowadays we cannot use it for free anymore. But nevertheless, it is a very important reservoir of fragmentation information. So basically, this is the look that you have in the database, and then you have your spectrum, what we just saw on the previous slide. And then you compare, do you have the same fragments or not, and then you can decide that, okay, so this one here, I have the very similar ones, and so that means that identification can be done, and the compound can be annotated based on the database. Also during the run, as you will see in the actual practicalities, in many times we have in-house databases and fragmentation spectral libraries, so that we can already during the analysis do it automatically on a computational form, so that the analysis already tells that, hey, this one has this and this fragments, it looks like this and this compound. So part of that can be already automatized, but already now how to say that you can never trust that automatic process. Every single identification has to be manually checked on the raw data level and curated, so, because those algorithms, they still make a lot of errors, so it still requires scientists there to make the final conclusion. So I hope I have now demonstrated you that the usability of mass spectrometry, but just to conclude here, why we want to utilize it in metabolomics. Naturally, for the reason to detect the maximum number of analytes, compounds, that can be possible with the chosen analytical techniques. So we have to also remember that we are relatively restricted in terms of the sample preparation and the analytical choices. So it's always a compromise between something. So we don't have the whole metabolome in a single analysis yet, but at least we try to maximize the number of compounds that we can see in a single analysis. Also, we should take such efforts that we can, so that the ionization could be as efficient as possible. So if you have something in your solvent, but if it is poorly ionized, it doesn't matter how high concentration you have, but if you don't manage to put it in an ionized form in the MS instrument, it will never be detected. So for that reason, the ionization is something that should be also taken into account in the analysis. And naturally, the aim here is to increase both the number of different components detected and also to increase the intensity of individual signals, so that they should ionize as efficiently as possible, because the more clear the signal from the component is, then you have better chances that it will be actually then picked in, for example, in this data-dependent MSMS analysis, and you will get a nice, clear fragmentation pattern that you can compare against the different databases then in your identification procedure. Then there are also huge range of various challenges. This course is not focused on mass spectrometry technology itself, so we don't go there in very detail, but just to mention that what we can encounter. So for example, ion suppression is one thing that we commonly see. What it means is that in your chromatographic separation, you might have a region in chromatography where a huge number of different compounds are eluding. And in some cases, part of those compounds can be such that they are very efficiently ionizing. So what happens is that they sort of suck all the electrons in the ionization process, and for that reason then some components that are not as easily ionized as these ones that are dominating, then they will sort of suppress the ionization efficiency of those that are co-eluding. So it's a little bit of a balance between both the chromatographic separation, but also then on the mass spectrometry side. So sometimes if in very unfortunate cases, you can have a situation that some dominant ions are really masking under something that is co-eluding there. That could be also very relevant. So that is one of the challenges.
So if I get it clearly, the ion the ion suppression means those compounds that you know are, that are eluting more efficiently, try to mask others that are not, amenable to ionization. And that is what ion suppression is.
Yes, so for example, in human plasma samples, we often see huge clumps of compounds called phosphatidyl, for example, phosphatidylcholines. And those are metabolites that are really efficiently ionizing. Luckily, typically in that region, we don't have that much other things that would be interesting. But if we suspect that we should see also something else, then what we should do is that we take the sample preparation process and, for example, get rid of the phosphatidylcholines, put them to some other solvent, have a SPE pre-column, for example, and purify the sample, and then run the sample again, and then we might see something that is masking under that clumps. And actually, we are just now discussing with Sarah's PhD work this kind of situation, so that we have a huge clumps of things that we have not seen earlier, and we are now thinking about what to do with those, and those are potentially ion suppressive constituents. So it's a very nasty situation actually, because before really getting rid of ion suppressive compounds and looking there, you don't know that beforehand. So it's something that it is in majority of the cases it is based on earlier literature, so that you know that this particular compound class is such that it can be masking behind more.
But can we say it could be beneficial in the sense that your compound of interest is actually emitting more, as in getting ionized efficiently? Could it be considered as advantageous, like in that case, when your compound of interest is actually getting ionized efficiently? Can you say the ion suppression effect could be beneficial in that sense, or because it is actually masking other compounds, you can say it is not actually reflecting what you're actually looking for?
Yeah, there are two sides. So of course, if your study focus happens to be those compounds that are super efficient ionized, then you are lucky. so of course you can then focus on those and gather the information. But it again depends a little bit whether that is really your study focus, if I understood your question correctly. So yeah. But on the other hand, then what the aim typically is that we nevertheless try to make as good chromatographic separation as possible, so that we wouldn't have that many compound classes overlapping. So I don't know any situations where we would be actually where it would be any sort of desired phenomenon, this ion suppression. So typically it is a harmful thing that is masking a lot of interesting things that could be there, but we don't know. Yeah. Yeah. Yeah, that's a very good question also. So in this kind of cases, typically what you need in the MSMS phase is that you have a data-dependent fragmentation, so that you have your compound A and compound B, and not to go there so that you fragment everything, which would then, that you still have two co-eluding compounds, and you have a group of MSMS fragments that you don't know which one is the source. In this kind of cases, the best thing is to have a targeted MSMS fragmentation, so that if your data analysis in the metabolomics workflow later on points towards these co-eluding compounds that you do see in your data, and both of them seem to be very interesting candidates for a biomarker, for example, then you could go back to your instrument, set up a targeted MSMS procedure, and each of these compounds would go to the MSMS fragmentation separately. And then you would get the fragmentation data for both of these separately, and then you can address and compare those against databases separately. And that is basically the only way of really separating and identifying those components. Even if they are co-eluding from the data analytical point of view, that is not that big problem if they nevertheless have different mass. So if it is still the different mass, the detector will say that, hey, here are two different compounds, they have a different mass, and they will be included in the data as different entities, even when they have the same retention time. But if the mass is different, then you can still separate those in the analysis and take those into fragmentation separately. Yeah. Actually, now I realize that actually I need to include in that slide still a separate approach for the targeted MSMS, because yes, it is true that targeted MSMS is data dependent, but these MSMS approaches that were listed here, they are the ones that are used in the whole metabolomics analysis. So when you set up your metabolomics analysis, you cannot have any targeted MSMS there yet when you run your samples for the first time, because you don't know what you have there. So for that reason, we use this data dependent or data independent when the instrument looks at the compounds and chooses what it fragments. But then later on, for example, in a case that you just described, that during the data analysis you notice that there are two compounds which are co-eluding, then you already know their mass. Then you can tell the instrument that, hey, let's look at these compounds in more targeted, precise manner. So that is yet another step here. If you are lucky, then of course these co-eluding compounds, they can already be there due to the data-dependent fragmentation. But if there are some minor ions, it might be that the instrument has not automatically collected those for MSMS fragmentation, and then you need to address them separately. Yeah, it's a forest of terminology, so it's good that you verify whatever is unclear. So. Yes, so we were going through these challenges in mass spectrometry. I mentioned ion suppression, and then also dynamic range is one of those typically listed, meaning that we have a very huge range of concentrations. But luckily this is an issue that especially the latest developments in mass spectrometry instruments, they can deal quite well, so that we can have concentration ranges from millimolar to attomolar even, or very huge ranges. But that's a issue that needs to be kept in mind at least. Also, challenges are related to co-elution of isomeric species, so that in a situation where we have the same molecular weight, and if they are not separated into chromatography, then it is a challenging situation. And also one aspect that is here included as a challenge, but also it's one of the greatest benefits of mass spectrometry-based metabolomics, is the discovery of novel metabolites without any database references for identification. So sometimes you might have very interesting and important compound that is clearly reporting an essential metabolic reaction of your study question, so that the mice who had only rye bread have this compound and it's really essential. But when you go through the mass spectrometry fragmentation pattern, you cannot find any matches in any databases. And then that poses a great challenge, that how can you then annotate that kind of compound? That is a big issue. Then you need several years of experience on really interpreting the data and calculating what kind of pieces and what atoms can you actually have in that component. So that is a challenge, but on the other hand also, I personally think that is one of the greatest benefits also, that you can make novel discoveries and you can find novel hypotheses when you run the data, so that you let the instrument tell you everything that can be seen in your samples. Then we can still look a little bit about sample sequence and quality control issues during LC-MS analysis. So these are very essential things for the reason that I already mentioned, that typically in metabolomics analysis our sample sequences can be very large. So for example, if we have a human dietary intervention sample, we can have tens of participants and different time points and different diets, and it's a very typical situation that several hundreds samples are analyzed on one go. And nevertheless, it is essential that all the samples that go to one metabolomics analysis are analyzed in the same time, as much as possible. Because whenever you need to then adjust the issues related to different analytical patches, then more error you are introducing, or you are likely to introduce more error to the data analysis. So here I show an example of a successful quality control procedure. Unfortunately, the situation is by far not always like this, but this is a good demonstration. So typically a quality control sample is such that when you have done the metabolite extraction of all your analytical samples, you take a small aliquot from all the analytical samples, let's say 10 microliters from each of the samples, and you mix those in one separate vial. And that mixture of all the analytical samples is then used as a quality control sample of your current metabolomics analysis. And this is a very good way to do, because then your quality control sample has all the same analytes that are present in the analytical samples, and also the matrix or the general composition of the compounds is matching with the analytical samples. And that is the most typical way to include quality control sample in metabolomics run. And for example here, this example is from a sample sequence where we had 236 analytical samples. Mixture was made, and also we injected some solvent blanks, and the quality control sample was injected after every 12 analytical samples. So altogether this analysis took 61 hours. So the instrument was running 61 hours without any interruption. And here, what you see here are the chromatograms overlaid from 20 quality control samples that are included during the analytical run. And as you can see, the chromatograms are perfectly overlaid. So this is a very comforting result from the initial quality control check, that your QC samples are perfectly overlaid, meaning that the signal from these samples is completely similar. And this is just a demonstration on how the sample sequence is typically prepared for metabolomics analysis. So in majority of the cases, when you have your Excel files filled with the sample names and information and so forth, then you should conduct a complete randomization so that they are injected in the instrument in complete random order. And if you are using filter plates for precipitation of the proteins, for example, here is a suggestion of where, for example, this set of samples is distributed on 3 different filter plates, and you have there also quality control and solvent blanks. And this is still an example of actual work list for a metabolomics run. So it's a little bit messy information, but typically in the beginning we have a priming sequence of quality control QC sample injections, and that is for the reason that we customize or accustom the column with the sample matrix that we are running. So something like 10 injection of QC samples typically in the beginning of the run. Those are not shown in this work list because it was too long. And then typically you have analytical samples in sets of 12 samples, and typically the samples are labeled only with running number, but then you have the code somewhere else. So it's not very advisable to have a lot of text and information in the actual sample label, but just simply the sample number is enough. And typically, what is the rule of thumb, at least in our lab, has been around 300 injections, and that is more or less the maximum 75 hours. But after that, then the instrument needs some maintenance and cleanup. So otherwise, it's not the signal is no longer reliable. And typically here then the QC samples are included after something like 10 to 12 analytical samples. And then here in the end, what you can see are different MSMS files, and these different MSMS methods can be then taken from different sample types and quality control samples and so on. So that can be always designed separately, that how can you maximize the information of MSMS analysis. And actually here I see I have used this terminology auto MSMS. So auto MSMS means the automatic MSMS analysis. So it's either the DDA or DIA that was included in the earlier slides. So this is just to let you know that also establishing the sample sequence and the analytical run for LC-MS metabolomics analysis is a little bit demanding and takes some time. And also here are a couple of situations where, as you can see, it's not perfectly overlaid. So for example, here on the left-hand side, there are chromatograms which are overlaid, but there is a clear shift in the retention times. So in this kind of situation, you know that there is obviously something wrong with the analysis, and you probably need to make some adjustments or even repeat part of the analysis. And on the right-hand side, then there is an example of another way of looking at the instrument is the pressure curve of the instrument during the run. And this is something that the analytical chemists typically keep an eye, because if the pressure changes a lot during the run, it means that there might be, for example, some kind of so that the column is not in a perfect condition, and it's an alert that if when opening the quality control sample during the run, if this kind of issues occur, then they need to be taken care. Then let's see, what is the time now? Okay, we can still start the remaining part of the workflow. And here in the data analysis part, we are going through the principles, but then later on during this course, you will be more accustomed to the actual practical way of the data analysis. But before going there, let's once more repeat that what do we have in metabolomics data? What is the structure actually? Because now I have shown you that we utilize the chromatographic separation and the mass spectrometry analysis, but how is that actually represented in the metabolomics data then? So what is collected there is all the information that we can get. So there is this mass to charge value, which is basically the molecular weight in ionized form. What is the mass to charge value for each component? What is the retention time? When did it come out from the column? And also the intensity. So this is arbitrary units account for the signal, meaning that how high amount of that compound we have in the analysis. And this is an information that we can of course look at on manually. This you will be also doing during the course, the lab work, so that you have the chromatogram, and when you click whatever location in the chromatogram, you can open the spectrum. And the spectrum shows all the ions that are eluting in that particular time, so that you can open at any of the locations. So in case when you are looking at the chromatogram, the x-axis is the time, the retention time, when each of these signals came out, and when you click on any particular individual retention time, then you can open the spectrum. And now in the spectrum, the x-axis is then the mass to charge value, so that it is telling how large compound you have. And the higher the peak, the more intense, more higher the concentration of that analyte is. So this is something that you need to be aware that you have two different ways of looking at the raw data. Either the chromatogram level showing everything, or then on the spectral level looking at individual peaks. And also in the chromatogram level, you can either look at what we call total ion chromatogram, what is like everything that is eluting, but in addition, once you have opened a particular peak or particular spectrum, like for example here is opened this particular retention time at one location of this chromatogram, you can take a single ion. So for example here, the ion 287 is chosen and taken the extracted ion chromatogram. So now with the extracted ion chromatogram, I can look at this individual compound, how it is eluting. So there in the total ion chromatogram, it might be something that is completely it's very crowded and it is behind there, but if I take the extracted ion chromatogram, I can see that with this molecular weight or this mass to charge ratio, where are signals eluting in the chromatogram. And in this particular case, you can see that with this same mass, there are 4 different locations where this kind of signal comes out. And the fact that we have good chromatographic separation makes it possible for us to distinguish these compounds even when they share the same mass. So this is the fundamental information, or like the knowledge that you have in the data matrix when processing metabolomics data. And it's good to already now get familiar and try to understand these different viewpoints of the data. And then when we go towards the metabolomics data, what do we actually collect there? We collect throughout the whole chromatographic run, every single spectral signal that can be seen. And this is a process that is done with the metabolomics software automatically, so that they take each sample, run through the whole chromatographic region and pick every individual signal and pick the intensity value for each of these signals. So these are the 3 different values that you will get for all of your analytes. Retention time, mass to charge ratio for the signal, and then the intensity values. And this is repeated throughout all the, let's say, 250 samples that you have in your run. And finally, those are put together in a data matrix, but obviously that slide was not here, or I cannot go back anymore. Okay, so well, in any case, that is the matrix that is collected for the data analysis. But before you go there, you should check the quality control samples that they are okay, and also open the total ion chromatogram of the actual samples as is done here. And you can see that there is a lot of fluctuation in the actual samples, which is of course a good thing, because it means that you do have metabolite and differences in the different analytical samples. And once you have opened these files and check that everything is fine, you don't have any huge deviations in your quality control or anything. And for example, as here you can see from the overlaid chromatograms that they are even when the intensity is different, still the same individual peaks are very well aligned, so it means that those are like retained during the run, so the analytical quality of the run is very good. But the intensity is different, and that is what we are expecting in metabolomics. So these are now then the raw data files that you push through the software that will do what we call the peak picking analysis, so that every single data file, every single retention time, all the different signals are then transported. This we already went through, so it's a procedure of peak picking, and why it's essential is that naturally in metabolomics analysis it's impossible to open all the files and examine all the signals individually, but you need to put that into a numeric form, into one entire data matrix that can be then explored in statistical analytical tools to find out the differences and so forth. And here there are various different freeware how you can do this peak picking. For example, MS-DIA that you will get familiar later on during this course. And also there are various commercial alternatives. So each of the vendors who produce mass spec instruments, they also have their own software for metabolomics analysis. But our lab has already several years utilized the freeware called MS-DIA. So I think this is now good location to stop this lecture, and we will continue from here then on Friday. Do you have any questions at this point? If not, then see you on Friday.